All right, so good afternoon, everyone. I guess it's afternoon when I record this, so I should also say good morning and good evening, depending on when you choose to watch this video, of course. This is the second lecture on molecular cloning and protein workup. In the first lecture, you guys probably focused more on DNA. You got a little bit of the DNA structure, you got a little background on cloning, and so on and so forth. And today we're going to focus more on the protein side of things. And before we get started, I want to say this is brought to you by CSberg. Of course, I hope that everyone's doing well during this difficult time. Now, in today's class, we will be discussing, and I will be providing an introduction to amino acids, peptides, and protein structure, CRISPR-Cas function and genome editing, and some more details on protein purification and analysis. Before we get into more detail with this stuff, I just want to say that with this class, what I'm going to try and do is give everyone a surface understanding of many different components, right? But we're not going to have the time to delve too deeply into all the different purification methods and all the different methods of protein analysis and so on and so forth. Now let's get started. Now, first and foremost, proteins are composed of individual amino acids that are attached together via peptide bonds. And you guys can see what a peptide bond looks like right here. This is considered a dehydration synthesis reaction, which makes a lot of sense because you can see that a water molecule is released during this bond formation. And at the end, of course, you get two amino acids that are attached together. One is on the left side of the blue line, and the other is on the right side of the blue line, shown here. An important point to note about this annotation that they're showing here on the peptide bonds, these R's shown here, you got an R here and an R here, they essentially represent any one of these highlighted regions. So you see there's a blue section here for glutamine, there's another blue section here for asparagine, and of course you got aspartic acid, alanine, for example, here has a yellow highlighted region, and histidine has a green highlighted region. So any one of these could be considered an R group. It's just shown as an R group here because it's a lot easier when you're discussing a peptide bond. Now, another important note about the peptide bond is that you'll see one side has a nitrogen and the other side ends with a carbon. So these are respectively called the N-terminus and the C-terminus, which makes a lot of sense. Now, in order to make a protein, multiple amino acids are attached together via these peptide bonds to create a long chain, right? And you can see that some of these amino acids carry a positive charge in their R group, some carry no charge and they're non-polar in their R group, some carry a negative charge in their R group, and some are polar in their R group, right? So again, just to clarify this, you'll notice that the top of all of these amino acids have exactly the same sequence that you see here and here, right? And that forms the peptide backbone, right? So that's where the peptide bonds are forming. And these R groups are the ones that are capable of interacting both with each other and with other things in their environment. So the question becomes, having all of this information, how do you go from multiple amino acids attached together in sequence to something that is a protein that can carry out some sort of function? And to discuss that, we're going to be looking at this really nice image that kind of describes how we go from a linear attachment of amino acids to something that is a functional protein. Okay, so on top here, much like we discussed, what we're looking at is individual amino acids as these circles attached together by these peptide bonds. That's the line that runs through them that holds them all together. And we see that there's an N-terminus here and a C-terminus here. This is what we call the primary structure. So this is just a line of amino acids. Now, in order to go from this line of amino acids to a functional protein, what needs to happen is that there needs to be structural changes. So you go from this line to something that looks like more of a folded group of amino acids, right? And the primary way this happens at the start is by transitioning from a primary structure to a secondary structure. Okay, in the secondary structure, hydrogen bonds, or non-covalent attachments between, well, shown here primarily as hydrogens and oxygens, start to bring together and develop a shape for this amino acid sequence. On the left side here, this is called an alpha helix. Okay, this is an alpha helical shape, so you see that it's kind of rotating around. And again, if you zoom in later on, or if you can clearly see this here, there's R groups here, much like the R groups that we discussed before from amino acids. And this is the peptide backbone. You'll see the nitrogen is attached to a hydrogen, and that hydrogen is hydrogen bonding to the oxygen of the carbon peptide backbone of another amino acid. So that's kind of folding it all together and creating this helical structure, which we call an alpha helix. Another form of secondary structure is called a beta sheet. In a beta sheet, you could just imagine that the protein kind of folds in multiple lines like this. That's a beta sheet. There's a parallel version and anti-parallel version, but we're not going to really focus on that. And again, same idea. It's held together by these hydrogen bonds. You see the nitrogen has a hydrogen here, and the hydrogen is hydrogen bonding with an oxygen that is on the carbon of the peptide bond for another set of amino acids. Right? And again, these hydrogen bonds hold everything together. So we've just gone from a primary structure to a secondary structure. Right? We went from something that was just a line of amino acids to something that has some form of a fold. Right? It looks like it's turning into something. Right? These amino acids are capable of interacting with each other. And then we move into the tertiary structure. So tertiary structures, without getting into too much detail, the primary forces that develop a tertiary structure, or additional folds beyond the alpha helix or the beta sheet, really kind of holding the protein together, those are nonpolar interactions, and disulfide bridges also help. So if you go to the previous slide, you'll see that there are some amino acids that have sulfurs. These sulfurs can actually form disulfide bonds under the right conditions, and those bonds are covalent bonds that kind of hold different parts of proteins together. So you could imagine if this section here of the amino acid sequence folds into an alpha helix, and this section here folds into a beta sheet, for example, right? You can imagine that nonpolar interactions between the R groups and or disulfide bonds might actually bring the alpha helix and the beta sheet closer together, so that you have a helical structure on one side and you have a sheet attached directly beside it. And that's additional structural information on this protein, right? So this protein has an additional structure to it now beyond just this helix and the beta sheet. And finally, in this case, there are proteins that are completely functional at this state, right? At a tertiary structure, you are folded, you can act on whatever it is that you need to do in the body, for example. But some proteins require quaternary structure. Quaternary structure, put relatively simply, is just multiple proteins having to interact together in order to enact some kind of function, right? So you just have multiple tertiary structures coming together. Here we have 4 identical tertiary structures here that can go ahead and do some kind of work. Now, before I finish this slide, and perhaps I should have started with this, but I think it's better late than never to mention this. Some people may have a relative misinterpretation on what proteins are, and hopefully this will be a relatively easy way to understand what a protein is. So, some people just think proteins are necessary for muscle building and so on and so forth. That is definitely not the case. So this amino acid sequence here that forms into helices and beta sheets and then comes together to form a structure, it is designed to produce a protein that carries out some sort of function. So what does that mean? Okay, so sucrase is a protein in the human body that converts sucrose to glucose, right? Now, sucrase needs to have its primary structure first, then it needs to fold into its tertiary structure, and then it needs to find a molecule of sucrose and convert it to glucose, right? So that is the goal of the sucrase protein. There is plethora of other proteins in your body that each have a different amino acid sequence, that is, the amino acids attached in different orders. So let's say you had 3 alanines here, another protein might have 3 histidines here, another protein might have a histidine and 2 alanines, and so on and so forth, in any combination you like. And each one of them has a different overall structure and a different output function, right? It does something else that your body needs. So for example, there are proteins in your liver that allow it to detox certain chemicals, right? There are proteins that are used, like I said, to go from sucrose to glucose. There are proteins in your muscles that can use energy and produce heat in order for your muscles to contract, right? And all of these proteins have their own individual amino acid sequence, secondary structure, tertiary structure, and relative function. Now, I was made aware in a previous presentation that there was a lot of information in the previous slide. So what I have here under protein structure is just kind of a summary for you on the really important and key points that we just talked about on the previous slide. And this is just before we move into understanding and discussing CRISPR-Cas function and how it works. Okay, so feel free to, if you need to pause the video, just look at this, write it down, jot it down, whatever you need to do. Okay, so yeah, let's discuss CRISPR-Cas. Okay, so most of you have probably heard of CRISPR-Cas, whether it is that you're in the sciences or outside of it. Some people give it a lot of flack. Other people like this system a lot. I am one of the people that likes the system a lot. I've worked on this before, so it's very, very cool. And I'm going to try to do my best here not to offend anybody that's been in the field for too long, while at the same time simplifying the concept so that a general audience can understand what CRISPR-Cas is and how it works. Now, CRISPR-Cas was originally discovered and understood to be an immune system in bacteria. Okay, so what does that mean? All right, so we know bacterial cells exist, and most of us think that bacteria are these evil creatures that try to get us sick all the time, but independent of that, bacteria have a lot of, let's just say, important things they do in the environment, right? And an important thing to note is that bacteria, much like human beings, actually have viruses that infect them. And these viruses are called phages. I'm showing you a phage on the left side right here. Now, these phages, simply put, are capable of interacting with the surface of a bacterial cell. This giant circle here is the bacterial cell, circle, rectangle, rectangle, circle. And they can inject their DNA, which is their genetic sequence, into the bacterial cell. Once they inject their DNA, phages are able to make all of the components they need to make other little baby phages, let's call them, right? And when enough baby phages fill up the bacterial cell, they burst out of the cell and kill the cell, right? So this is pretty bad for bacteria, if you were to think about it, because you're getting infected with this phage, and then your endpoint is death, right? So it makes a lot of sense that bacteria over time have evolved mechanisms to try and protect themselves against phages that try to infect them, against these bacterial specific viruses shown here. Now, CRISPR-Cas is one of these defenses, and I'm going to try to go over this relatively quickly. Okay, so what you're looking at the top is the DNA sequence, and it's an annotated DNA sequence here of a CRISPR-Cas system. So a CRISPR-Cas system has a CRISPR array, also known as a clustered regularly interspaced short palindromic repeat array. You can search that on Google to get more details on it. But the CRISPR array is essentially composed of repeat sequences and spacer sequences. The repeats here are the light gray, dark gray boxes, and the spacers all have different colors. Repeats are all identical sequences to each other, so all of these repeats are exactly the same sequence. And the spacers are usually, which make it easy here, they're all different sequences. And each one of these sequences can interact with a piece of foreign DNA. So what I mean by foreign DNA is, let's say phage DNA is foreign DNA. It's not DNA that's from the bacteria, right? So the spacer can interact with that phage DNA. From the DNA workup that you guys did in the previous lecture, if you transform a plasmid into the cells, it's the same idea because it's foreign DNA. If there's a spacer that matches the plasmid, the spacer can interact with the foreign plasmid DNA. It's fairly straightforward. And the second component of the DNA here are the Cas genes. The Cas genes code for proteins. So we discussed before, these are these individual proteins that are folded in a certain way and can do something in the system, right? So in order to provide protection against foreign genetic sequences, like a phage genome, for example, the CRISPR array is transcribed, and you end up getting this RNA sequence. And from your first lecture, you guys probably learned that RNA is made from DNA, so that makes sense. You're transcribing your DNA sequence, you're getting RNA out of it. And you'll see here that the repeats are the black sequences, they get this looped formation, and the spacers are, again, the same colors, right? There's a processing step that we're not going to talk about, but the result is that you get these mature CRISPR RNAs that they're called, and the mature CRISPR RNA carries the spacer, and it carries a little bit of the repeat on either side. Now, depending on which CRISPR-Cas system we want to talk about, we're only going to focus on one later, but just to give you a little bit of a broad overview, some CRISPR systems have only one Cas protein that they need to do what they need to do. Other CRISPR systems have multiple Cas proteins. In this figure, you have multiple Cas proteins that have come together to interact with the CRISPR RNA here. And again, you can see you've got a little repeat on the left, you got a little bit of the repeat on the right, you got the spacer in the center, and these Cas proteins are interacting with the CRISPR RNA. Once this interaction occurs, the CRISPR RNA spacer can then go and anneal to the DNA area that it matches on the phage, right? So the moment this annealing occurs, it tells the Cas proteins that the cell has recognized a foreign piece of DNA, and this foreign piece of DNA needs to get destroyed, right? Because it might replicate in the bacteria and then kill the host. So the Cas proteins go in and start to cut the phage DNA at different parts, in different places, right? Depending on the system, sometimes it's just one cut, sometimes it's multiple cuts. But the point is that once the phage DNA here is destroyed, the phage no longer can infect the bacterial cell. So in that regard, the CRISPR-Cas system was thought to be a really cool and interesting defense mechanism. Now, CRISPR is an adaptive immune system, but we're not going to talk about the adaptive component of this. I think if you're interested, I urge you to look that up. It's actually fairly cool. But in essence, this is all that CRISPR is. You get the CRISPR RNA formed, shown here. The CRISPR RNA interacts with the Cas proteins, and when a CRISPR RNA recognizes a piece of DNA, and I'll get into more details with this later, the Cas proteins cut the phage DNA, and the DNA no longer can infect the host. Now that we've discussed what CRISPR-Cas really is in its environment, we can go into, I guess, what I would call the Houdini aspect that most people like to talk about with regards to how CRISPR-Cas is used. And that's the idea of using CRISPR-Cas for genome editing, right? You want to edit human cells in tissue culture, you've probably heard CRISPR-Cas9. You want to edit E. coli cells, CRISPR-Cas9. You want to edit Streptococcus pyogenes, CRISPR-Cas9. You want to edit, I don't know, a pig, CRISPR-Cas9, right? So, okay, let's discuss what that is, what it means, and how researchers have taken this immune system in bacteria and are now using it to try and make mutations in different cell types, right? And what I want to mention here is that there's a lot of discussions about how CRISPR-Cas is used, or how CRISPR-Cas could be used in inappropriate ways, and so on and so forth. But one thing that's very, very important to note is that when looking at CRISPR-Cas and from the lens of research, it is an incredible tool, right? It allows us to edit the genomes of all the things that we do research on, so we get a better understanding of how everything works inside individual cells and inside animal models, for example. So that's an incredible thing. Of course, with the animal models, we have to be a little bit careful, but you get my point. Now, we talked about CRISPR-Cas, and I mentioned that there are some CRISPR-Cas systems that only use one Cas protein to do what they need to do. CRISPR-Cas9 is one of those, right? So what you're looking at here are these two blue blobs. I know they're separated, but it's actually just one protein. These two blue blobs represent Cas9, right? And that's just one Cas protein that can do the whole thing. It can interact with the CRISPR-RNA, it can interact with the DNA, it can cut the DNA, right? So it can do all of these things with just one protein. It's excellent. On top, you're looking at the annotation of Cas9 from the organism Streptococcus pyogenes. Right, so we're not going to get into the details, but this is generally the annotation that you would look at if you're looking at this type of CRISPR-Cas9. So there's a C-terminal domain, there's a RUB-C section that's split, there's a H and H domain, and there's a helical domain here, helical domain 1 through 3. Two of the important domains here are the RUB-C and the H and H. So H and H is a nuclease, RUB-C is also a nuclease. If you guys know what a nuclease is, it's basically something that can cut DNA. So it makes a lot of sense. Cas9, a protein that's supposed to cut DNA, has two different DNA cutting domains. On the picture here, one of them is above, one of them is below. So you see RUB-C here, H and H on top of it, and it's annotated as scissors here. All right, so I want to go into a little bit more detail of how Cas9 is able to interact with its target and how it's able to cut DNA very specifically. And after we discuss exactly how Cas9 works, because it's being used a lot in research nowadays in a lot of different organisms, then I'll discuss a little bit about how you can make mutations with it. All right, so what you're looking at here is you're looking at the CRISPR RNA. Here it's an sgRNA. It's a single guide RNA. We'll just call it a variation of CRISPR RNA here. But this guy here is the spacer area, the dark orange area on the CRISPR RNA that kind of looks like it's interacting with the DNA on top. And then this is the part of the repeat section, right? Part of the repeat section is interacting with Cas9. The second thing to really note here is that there's your target DNA. So this is, let's say, the foreign DNA, and you've got it annealed perfectly here, but as it kind of goes into the mouth of Cas9, it is unzipped. So you see this unzipping action on the top and the bottom strand, and then it zips back together towards its annealed form, right? So one of the things that this CRISPR RNA and Cas9 together do is they open up the DNA sequence in the area where the spacer, shown here, matches the DNA shown on top, okay? And before I told you in a very simple way that the spacer guides the Cas proteins to the target and it cuts, that's correct, but it's not perfectly correct. So there are two signals that CRISPR-Cas looks for in order to understand whether or not it should be cutting a piece of DNA. The first signal is the spacer sequence. Well, we'll go over them in reverse order and then I'll explain exactly how it works. So the first signal, which we discussed before, makes life relatively easy, is the spacer. So you see here, if the spacer is a good match for the target DNA, that's the first indicator that this is probably foreign DNA, right? And that kind of gets Cas9 all worked up and excited. The second signal that's extremely important in CRISPR-Cas9 targeting is this PAM motif, also known as a protospacer adjacent motif. Okay? Now, PAM for Streptococcus pyogenes is NGG. This is a nucleotide sequence. N is any nucleotide, and GG is two guanine. So guanine, guanine. Okay? Now, if Cas9 itself, the protein, sees the NGG on the DNA strand that it wants to target, that is the second signal. Okay? So the first signal is the RNA interacting with the DNA to form a hybrid. The second signal is Cas9, the protein, interacting with this NGG domain to tell it that it is a correct target, right? Now, the reason why I said I'll explain something later is because Actually, the way we think this works is that the first thing that happens is Cas9 kind of tries to look for an NGG domain first, and then kind of slips in this CRISPR-RNA to interact and see whether or not the DNA sequence matches. But irrespective, in order for you to get cleavage, you need both signals. You need the CRISPR-RNA in the DNA, and you need the Cas9 to interact with the DNA also. When both of those signals are sufficient, Cas9 creates a double-stranded break in the target DNA by cutting both the top and the bottom strand accordingly. Right, so like I promised, how do we go from this CRISPR-Cas9 targeting system to making mutations in a cell, for example? Okay, so let's say in our imagination that we had Cas9 placed into HEC293 T cells. Okay, HEC293 T cells are a tissue culture cell line, human tissue culture cell line. We'll just leave it at that. Makes life easy. Let's say you've got Cas9 in there, and you've made a guide RNA that targets a piece of DNA in HEC293T. Life's fairly straightforward. So Cas9 goes ahead and does what it does best, right? It finds the little piece of DNA that you want it to cut and makes a double-stranded break, and you got one piece of DNA on the left side and one piece of DNA on the right side. Life is good so far. We understand how this works. Now, what's interesting about double-stranded breaks is that cells have mechanisms to try and repair double-stranded DNA breaks, right? And you can kind of extrapolate this, right? You've thought about it. You know, you go out in sunlight, people tell you UV damages your DNA, but you don't have any adverse effects usually. And that's because your cells have DNA repair mechanisms, right? So when they see a double-stranded DNA break, when a cell sees a double-stranded DNA break, the first thing it's thinking about is, okay, how do I go about fixing this, well, these two pieces of DNA that are supposed to be together, right? Now, the method on the left is the easier method of the two, and I'll just describe that really quickly. So it's called non-homologous end joining. To make your life really simple, when a cell sees two pieces of DNA and conducts non-homologous end joining, it's just going to take the two pieces and stick them back together, right? And it's using proteins to do that. These proteins are specific for, holding two pieces of DNA, chewing back certain pieces, and there's a lot of procedures with that. But the point is that when the cell tries to put the two pieces of DNA back together, because it doesn't really have any kind of template, it starts to make some mistakes, right? So it can make an insertion mistake, that is, it adds a couple of extra nucleotides in the DNA shown here, a small deletion, so it deletes a few nucleotides in the DNA shown here, and sometimes you just get a random mutation forming, right? So in the form of non-homologous end joining, you could imagine that, it's a fairly simple method for us to make random mutations in a cell. So what you can do is, you get Cas9 in there, it targets the DNA, you let the cells recover, you send that stuff for sequencing, and you ask the question, okay, well, how are these cells acting, and what's the mutation that they have in the area that we targeted? Fairly straightforward, very easy. The only problem here is that these mutations are random, right? So you don't know what kind of effects they might have in the sense of how much you can really know about what kind of effects they're going to have. The second method is a little bit more specific. So in the second method, what a researcher would do is they would get their double-stranded DNA break, but they would also have their cells transformed with, or their cells would also carry a double-stranded piece of DNA called a donor DNA or template DNA, right? And what that donor DNA is, it basically looks very similar to the area that CRISPR-Cas9 targeted, but it has very specific differences that the researcher has decided on, right? So let's say a sequence was ATG, and I gave the cells a linear piece of double-stranded DNA that had GTA in place of the ATG, right? Now, through homology-directed repair, which is another repair mechanism, what the cell can do is, instead of just sticking the two pieces of DNA back together, what it can do is look in its environment to see if there is a double-stranded piece of DNA that looks similar to the DNA that's been cut, right? And when that happens, what the cell can do is take the donor template DNA that we have provided and stick it into the area where the cut has been formed, right? And in this way, a researcher can more specifically decide what kind of mutation they're forming in a specific gene in a cell, right? So non-homologous end joining is, let's say, more random. And when you use a donor template to do homology-directed repair, it's more specific. That's essentially it. If you wanted to do this in bacterial cells, they also have a non-homologous end joining system, and you can use homologous recombination, which is kind of the same, but a little bit different than eukaryotes, but the general idea is the same. You cut and then you use one of the two mechanisms to try and make the mutation that you want. Okay, so in light of the fact that I essentially explained that you'd have Cas9 inside cells and you'd have a guide RNA or a single guide RNA inside the cells, which is a variant of the CRISPR-RNA, and they would come together and target your DNA. One of the things that's really important is for you guys to know what the sequence of the guide RNA is, right? So let's say, down the road, 1, 2, 3 years, you want to go into research, right? And you want to use Cas9 to target something, right? Under normal circumstances, what you're going to have is you're going to have a plasmid that's carrying, ooh, this is interesting. You're going to have a plasmid that's carrying Cas9 on it, and it's also carrying the sgRNA. This is all going to be in DNA, right? Cas9 will then be transcribed into RNA and translated into protein, and the guide RNA sequence, well, I guess it would be guide DNA in that case, would be transcribed into a guide RNA. So those two things would come together and they would go ahead and target. This is what the guide RNA looks like in RNA form, okay? And importantly to note, these ends here, these 20 ends represent the spacer. So if you're a researcher and you want to target a specific piece of DNA, you find the DNA, you find the sequence that you're interested in, you take that sequence and literally put it where these ends are, okay? And a very important point on picking a sequence is you got to make sure that at the end of this end, the last N, right, in the DNA sequence, it says NGG. Notice in the RNA it says GUU, so it's not related. But in the DNA sequence that you got to target, it's got to be a perfect match on these ends, these 20 nucleotides here, and then it needs to be followed by NGG. If you have that, and you've put this guide RNA in, Cas9 will cut what it is that you want it to cut, okay? And also I'm showing you guys this because you get to see what the guide RNA sequence looks like, right? So if you want to know the exact sequence, this is it. So that concludes the rather long discussion on how CRISPR-Cas works. There's just a few points here. I'm calling it a type 2A CRISPR-Cas system. That's what CRISPR-Cas9 is, a type 2A CRISPR-Cas system. I alluded to this before that there are many different types. You guys can go ahead and search online different types of CRISPR-Cas systems. It'll show you other types. And what we learned is that CRISPR-Cas9 is composed of Cas9 protein and the guide RNA in essentially the end form. The complex uses RNA, DNA, and protein DNA interactions in order to cut DNA, forming blunt end cuts. That's just a double-stranded DNA cut. And we also discussed how the simplicity of CRISPR-Cas in genome editing makes it a great tool to make mutations in many organisms. And this conversation that we just had is a great segue in many forms to discussing strain engineering. Okay, so what is strain engineering? Strain engineering is the idea of being able to take, let's say, a bacterial strain or a tissue culture cell, and that wouldn't be a strain, but you get the point, a tissue culture cell, and mutating it in some way, shape, or form, or getting it to express something that it's not supposed to express, right? And I'm showing you the three of the methods that, two of them you've already talked about, one we're going to talk about next relatively quickly. So CRISPR-Cas genome editing, it's easy, I think, for you now to kind of imagine how you could use that. If you could target DNA, if you could put in whatever you want, or you can take out whatever you want in theory. The idea there is that if you have E. coli, for example, and you want to make a knockout, you want to remove a gene, you can use CRISPR-Cas to cut and put in something else, right? If you want to overexpress something. You can use CRISPR-Cas9 and change a promoter sequence. I don't know if you guys have talked about promoter sequences, but the general idea of a promoter sequence on DNA is just something that helps with the expression of a DNA sequence, right? So if you wanted to overexpress something, you could use CRISPR-Cas to make a mutation in a promoter region and then get overexpression of something that you would want, right? So this is a very straightforward way of you making a edit in the genome of a host so that it can act the way you want it to, right? In the previous class, you guys probably talked about plasmid engineering and cloning. Generally the same idea here, right? Like if you don't want to use CRISPR-Cas genome editing and put a new piece of DNA into the genome, you can express this piece of DNA that you're interested in off a plasmid. So you transform your cells with a plasmid. Let's say you've got E. coli, you transform it with a plasmid, and this plasmid is going to be expressing your gene of interest. And again, with a plasmid, you can put a different kind of promoter upstream of it, and so on and so forth, and see how that works inside your cell. So it's pretty straightforward. And the last part here is directed evolution that we're going to talk about today. And directed evolution is very interesting. It's related to strain engineering, but it can also be used for evolving a specific gene, right? So the next slide, I'm going to talk about evolving a specific gene, and then I'll kind of describe how you could use directed evolution to evolve a strain, which would fall under the section of strain engineering. Okay, so directed evolution of a gene, a piece of DNA that codes for a protein, right? So you take a gene of interest, let's say. You really like the gene, you want to make it better at what it does. Perhaps this might be a bad example, but for the sake of argument, this will be fairly straightforward based on what we discussed before. So let's say you got the sucrase gene. You want to make it better at turning sucrose into glucose, right? Because you eat a lot of sucrose. So you're like, I'm going to make this gene better at turning sucrose to glucose. So you take your sucrase gene and you can do something called mutagenic PCR. So PCR, polymerase chain reaction, is a way for you to amplify a piece of DNA, right? But if you do mutagenic PCR, what you're doing is randomly mutating your gene of interest, okay? And you can make random mutations by using a polymerase that has low fidelity, for example. Or you can change the types of nucleotide concentrations that you have inside your PCR mixture. So let's say you have more A and T, less G and C. That's going to force the polymerase to add incorrect nucleotides across the sequence as it's trying to amplify. So after your mutagenic PCR, you end up with a gene library. So each of these genes are slightly different from each other based on the random mutagenesis that you created. You can put all of these genes into a plasmid and transform them, let's say, into E. coli, right? So you got your E. coli expressing gene library. Each E. coli cell, each colony of E. coli that you pick technically has a different gene expressed off its plasmid. So thus far, we just have a randomly mutated gene, and each E. coli is expressing a differently randomly mutated gene, right? Now, we could take that and conduct activity assays after we purify the protein output of this mutated gene, and we're going to talk about that later. And if you find one protein that does a much better job at converting sucrose to glucose, you can take it and put it right back into your E. coli strain. In fact, if you have some stock of this, you could just know that this is a strain that you're interested in. Now, what's interesting is if you want to make the gene even better, or just get to the absolute maximum best of converting sucrose to glucose, you can take that gene again, do another mutagenic PCR run, make another gene library, and go at it over and over again. And the idea here is that every single time you run this system, you're making your sucrase gene better, right? So the sucrase gene codes for a better sucrase protein that's capable of converting your sucrose to glucose faster, right? Let's say that's what you're looking at. So that's directed evolution if you're doing it for a gene. You can also do directed evolution for an organism itself. So let's say you want your E. coli to grow under certain acidic conditions, right? Let's say it's very acidic. What you could do is throw a strain of E. coli that you have in a certain level of acid and slowly over time decrease the pH so it becomes more and more acidic, right? And what will happen is, because the cells are trying to survive, there will be random mutations occurring, obviously, when cells replicate their DNA. These E. coli cells replicate their DNA. And if any of these cells ends up being better at surviving in acidic conditions, it'll outgrow the rest of the population. So what you'll be left with in a flask is E. coli cells that are better at growing in acidic conditions. And what we can do there is basically take the E. coli cells, sequence it, and then look at the kind of mutations that you have, what proteins were mutated, what parts of the DNA were mutated, and how you think that might be impacting the ability of this E. coli strain to grow better in acidic conditions, right? And that would be the version of directed evolution for strain engineering, which is what we were talking about before. Okay, so in part, before this slide, what I was talking about was the idea of, let's say you wanted your sucrase gene to be better, you purify that protein, and then you do some activity assays. But, saying purified protein, there's a lot to it, right? So one of the things that we're going to focus on now for the next section of this talk is going to be, well, how do we purify and analyze protein function, right? We're going to talk about exactly what you need to know. So the conversation here is going to be focused on proteins extracted from whole cell lysates. So the idea here is you got your E. coli strain. You want to get at one specific protein in your E. coli strain, even though it's expressing a plethora of proteins, right? So how do you get to that specific protein? And then after you've gotten that separation, after you've got that purification of your protein, specifically that structure that is what you want, how do you figure out what its structure is? How do you figure out what it's interacting with in the cell? Perhaps what its ligands are, so on and so forth. And that's going to be the main focus for the next part of this talk. Now, to talk about this stuff, there's going to be 5 sections, I guess, that we're going to review. And I want to make this clear. There's a lot more that you can learn about, right? Just distilling it down to some interesting methods that people use, but there's a lot more that you can do, right? There's a lot more that you can do in terms of protein purification analysis, right? So today we're going to talk about affinity purification, and that is, I want you to think of it as a starting point for you to get a purified protein, right? So your protein of interest, you want to have it relatively pure. Affinity purification is the first thing you're going to do. Next, you do gel chromatography or fast protein liquid chromatography, which is basically an updated version of gel chromatography. This can help you get an even more pure version of your protein of interest, right? Like now you have a high purity sample of just your protein. Next, if you want structural information, you can do crystallography or NMR. NMR is nuclear magnetic resonance in order to get structural information on the protein you purified. If you want to learn about protein-protein interactions and protein-ligand interactions, you can do isothermal titration, calorimetry, or surface plasmon resonance. There's other stuff you can do, of course, but those are the 2 that we're going to talk about today. And at the end of this, and I won't be talking about this in this presentation, but I will be providing a link. There'll be a discussion on enzyme kinetic assays, right? So enzyme kinetics assays, it's a way for you to determine the kinetics of your protein. So remember when I was talking about sucrose to glucose, for example, it's like, what is the rate at which sucrose gets converted to glucose? How many sucrose molecules can one protein of sucrase interact with, and so on and so forth. So that's the kinetics of the stuff. Enzyme kinetics can be very complicated, and there's a lot to it, and we won't have enough time to get into it, but the link that I provide at the end of this slideshow, if you go to it, does a fairly great job of explaining exactly how enzyme kinetics work and how you can use them. Without further ado, let's start with affinity purification. Now, in this slide deck, what I will be talking about is his tag purification specifically. There are other ways to purify your protein. We're just going to talk about his tag. It's relatively straightforward. So let's say you've got yourself a plasmid, right? And your plasmid's been transformed to your cell. Imagine that there's, I don't know if you guys can see the oval here, but your plasmid's been transformed to the cells, and it's expressing your protein, right? And this guy right here is your protein. It's called a tagged protein. I'll explain what the tag is. And of course, the cell is obviously expressing other proteins that it needs for its function. So if this is E. coli, it's expressing a bunch of other stuff that it needs to go on about its day and replicate. So the problem here is, how do we get at our protein only and remove all of this other, well, I want to call it crap, but like all this other stuff that we're not interested in, right? And one of the ways to do that is to tag our protein, right? So a way to tag a protein is called a his tag, right? And what that means is on your plasmid sequence, as the plasmid is like a little, imagine it as a little circle, at either the N terminus or the C terminus of what the protein will be, right? You can add a nucleotide sequence that codes for 6 histidines, right? Histidines are amino acids. So the idea here is that your nucleotide sequence, when it's translated, when it's transcribed and then translated, what you'll end up getting is a protein that either at its N terminus or at its C terminus will have 6 histidine amino acids, one after the other, right? Because this tag is relatively small, it usually doesn't have an effect on the overall structure of your protein, so usually you don't have to worry about that, but sometimes you can complicate things. So let's assume that we've expressed this protein of interest here that we liked, and it's got these 6 histidines, right? In this case, it looks like they've done it so that it looks like it's on the N terminus and the C terminus. Let's just imagine that it's only on the N terminus. Once we have this tag, what we want to do is take it and somehow purify just our protein of interest. Now, what's interesting about multiple histidines is that they can interact with nickel, okay, nickel beads. And so what we can do is something called nickel affinity chromatography, where we run something through a nickel affinity column, right? So the first step here is you've got your cells with your protein. You have to lyse your cells, which means kill everything. You can use sonication for this, which is a method of basically ripping apart the membrane of your E. coli cells. And that allows you to access everything on the inside. So now you've got all of the internal proteins mixed together, right? You can take all these internal proteins mixed together in a buffer and throw them onto a nickel affinity column. And what the nickel affinity column will do is specifically bind to your his tag protein and not really to anything else inside the cells. Now, it should be noted, right, there could be some things that may have a couple of histidines on them, and they might be exposed on the surface, which means the nickel affinity column might also bind a few other things, but overall, when you do this, you pretty much clean up most of the debris that you are not interested in, right? So you run a couple of washes with buffers, and you get rid of anything inside the cell that you're not interested in, and then you do an elution run, and you basically just elute your purified his tag protein, right? And that's shown right here. So at this point, it's great. You've got a relatively pure, but not perfectly pure, his tag protein. You can move on to the next step, which is running it through a column in order to purify it even further. All right, so this is essentially the second column you're going to run this protein through. The first column had nickel beads in it, so you had nickel on them. This column is just going to, let's make it relatively easy. It's going to have these, the way the figure shows it here is it'll have these little beads on it, right? And notice how these little beads have different like crevices on them. Some of the crevices are a little bit smaller, some of the crevices are a little larger. It's called a resonance column matrix. And so what you're going to do in gel chromatography is essentially take this semi-pure sample that you have and run it through this second column, okay? And what's interesting about gel chromatography is that the column will separate proteins based on size. So any other proteins that came down through your nickel affinity purification, they're very likely to be of different size, right? Different hydrodynamic radius. I'm not going to get into the specifics of that, but you guys can definitely look that up to know what it means. And based on that difference, when you run things through the column with the force of gravity, things will separate, right? So what you're looking at on the right here is if a protein is significantly larger, it cannot make its way into these little crevices on the beads, right, on the Sephiros beads that we have here. So it essentially just goes around everything and elutes very, very quickly, right? So the first thing that's going to come out of the column is the relatively larger proteins, right? As the proteins get smaller and smaller, the crevices that they can access become larger and larger. Notice how this green one can actually enter all of the crevices on these beads, whereas the red one can access a lower number of them, right? So when you're running this column, what will happen is bigger proteins elute first, and then let's call them medium-sized proteins, and then small proteins, and then tiny proteins, and that's the order in which they're going to elute. So because of the ability of gel chromatography to separate based on size, what you can do is find where your protein would be based on the expected size that you know your protein is supposed to be, and then know exactly when it's going to elute, right? So what that'll do is allow you to get that final level of purification that you need, so that it's just your protein in the mixture and there's nothing else in terms of debris in that mixture. Now, fast protein liquid chromatography, to make it really, really easy, is just, let's say, just researchers being impatient, right? Let's say that. So a fast protein liquid chromatography, you essentially add pressure at the top of the column to make the liquid run through faster, right? So instead of, adding your mixture of proteins on top, your relatively purified protein mixture on top, and then waiting until the force of gravity does its job to run it through the column and separate everything, what you can do is add pressure on top and force the liquid through the column much faster. Of course, for anybody who's done physics before, you don't want to make the pressure too high because you might burst the column, but that's the general idea of fast protein liquid chromatography. They essentially do the same thing here. So all you're doing is you're further purifying your protein by separating it based on size, right? And that ends up giving you a purified version of your protein. Okay, so you have this purified protein. Let's say that the first thing you want to do is determine the structure, right? There are two ways to do that. One is nuclear magnetic resonance spectroscopy. The other one is X-ray crystallography. We're going to talk about NMR first. We'll talk about X-ray crystallography afterwards. I'm going to try to make NMR less physics and more science, but we'll see if that's even something you can do. Okay, so the general idea behind how NMR works is that if you have an atom that has an odd number of protons or an odd number of neutrons, right, that atom has a certain level of angular momentum. Now, let's not get into too many specifics here, but if you guys took first year physics in undergrad, for example, you'll know that if there is a charge that's moving, right? Let's say in a circular motion. If a charge is moving, it creates a magnetic field, right? And that's essentially the way you should think about atoms when you're doing NMR, right? Each atom, let's say carbon-13 has an angular momentum. So each atom of carbon-13 is kind of like a little magnet, okay? And you guys, even in high school, you see that if you put like two magnets in the right orientation, they stick together. If they're in opposite orientations, they try to repel each other, right? So the idea behind nuclear magnetic resonance spectroscopy is that, let's say for carbons, let's just talk about carbons right now. Let's say you've grown up your protein in E. coli cells that are exposed to carbon-13. So what these E. coli cells are doing is essentially putting carbon-13 all over the amino acid sequence of your protein of interest, right? So your protein is just full of carbon-13. Now, if you purify that protein and throw it into an external magnetic field, you'll see here this is no magnetic field, this is an external magnetic field. What will happen is each one of those carbon-13 magnets will want to align with the direction of the magnetic field, right? Here they're just pointing in any whatsoever direction because there's nothing really forcing them to move one way or the other. But when you put it in an external magnetic field, you notice that some of the carbon-13s are pointing in the same direction, some of the carbon-13s are pointing in the opposite direction. One way to kind of understand this is that the way each carbon atom in the protein feels the external magnetic field is different based on what is in its surrounding environment, right? This protein is folded, right? So you could imagine that some carbons are more shielded from the magnetic field than other carbons are. And that's just because they have more atoms in their surrounding environment, right? That's why there's variation here. That's why some are pointing in the direction of the magnetic field, some are pointing against it. A point to note here, though, is that all of them would be in a lower energy state. That means they would be happier, right, if they were pointing in the direction of the magnetic field. It's that simple. Now, if you wanted to figure out where these atoms are located, right, you would use NMR. In NMR, you basically take your sample and you stick it in an external magnetic field. So these two plates you could imagine are creating that external magnetic field. So now you've got this kind of look, let's say, inside your test tube. Some of the carbon-13 atoms are pointing downwards, some of the carbon-13 atoms are pointing upwards. And the ones that are pointing downwards have a higher energy than the ones that are pointing upwards, right? The ones that are pointing upwards are happy, and happy usually means lower energy, right? Unhappy means higher energy. They're very angry, they're very jittery, they're very aggressive. You can think of it that way. Now, once we have our sample inside the tube with this magnetic field on the outside, if we blast our tube with a radio frequency that's the right strength, right, the right frequency, what we can do is get some of the atoms that are pointing in the direction of the magnetic field to switch and point in the opposite direction, right? So now they're pointing against the magnetic field, which is not what they're supposed to do, right? But the only reason they do this is because this radio frequency can be absorbed as a source of energy, and it can allow the carbon-13 atom to go from its lower energy state to its higher energy state, okay? Now, if we then remove the radio frequency, what eventually happens is this higher energy carbon-13 atom eventually loses that energy, right? It releases that energy and then goes back to its regular state. Now, that release of energy is computed, right? And that gives you information about the location of that specific carbon-13 atom, right? So I think hopefully you can kind of get an idea of how NMR works in a very general sense, right? By constantly flipping the direction of each individual atom or multiple atoms in solution, right, you can determine where each of these atoms are located in the solution, right? And you can start to formulate, in the context of just carbon-13, a 1D model about where each of the carbon-13 atoms are located, right? So, if we move into an NMR spectrum, let's say you want to get the full spectrum, you want to get that 3D structure, you don't want the 1D structure, right? You want additional information. There are 3 atoms that you can use in your protein. These are found in amino acids, right? The hydrogen, the nitrogen, the carbon, right? That are all have angular momentum. So they all have spin, which means they all have their own little magnetic field. Now, you can imagine if I have information about where the carbons are located, where the hydrogen are located, and where the nitrogen are located on this amino acid sequence, on this protein, right? I can get information about the 3D location of each one of these atoms, right? Because now I'll have, like, let's say I have a carbon here, and I'll have a nitrogen here, and I'll have a hydrogen here, and I'll know that they always present themselves in the same position. I have now learned something about the backbone composition of that specific section of the protein. You obviously need to solve this, and it can be very difficult to do that, but the point is that this is how NMR allows you to determine protein structure in solution, right? That's the general idea. Now, let's move into X-ray crystallography. So let's say you wanted to use X-ray crystallography to determine the structure of your protein. Now, for anybody who's a crystallographer out there, this is not going to be very detailed, so I really hope that it doesn't, doesn't make anybody too unhappy. But the general idea with X-ray crystallography is that the first thing you need to do is have your purified protein in a certain buffer that allows it to crystallize. So you can imagine it as what crystal would be is each one of your proteins in exactly the same conformation all together. They're packed together and they form a structure. That's an easy way of thinking about it. It's not trivial, it's actually very difficult to do, but once you have the right conditions and you have a crystal, you can move on to doing X-ray crystallography. And what you do there is essentially shoot an X-ray beam at the crystal. And what the crystal will then do is diffract the X-ray beam accordingly, right? And that diffraction pattern tells you a lot about the location of, or the electron density, to be more clear. It tells you a lot about the electron density of that specific angle at which you're shooting the X-ray at the crystal, right? So what you can do then is change the angle that you're shooting that X-ray beam at the crystal and get more information about the electron densities at different angles with which you are looking at the protein. Does that make sense? Hopefully. So the general idea is by getting electron density information, you can kind of get an idea of where the atoms are located in this protein structure. Once you have all of the electron density information, and there's a lot that goes into this, you have to worry about phases and phase problems and so on. It's a whole shebang. But let's just say it's that simple. That's when you have to start trying to solve the structure. Two of the methods that you can use to solve the structure are, one, you can use multiple isomorphous replacement. The idea here is you're soaking your crystal in heavy atoms, and then you're shooting your X-ray at this crystal that has heavy atoms in it. You compare that to the X-ray without the heavy atoms. And the heavy atoms basically give you an indication of exactly where they're located, so you can get an idea of how to orient yourself on this protein. The other method is molecular replacement. If you're lucky and let's say you're getting the structure of a homologous protein, something that somebody already has a structure of, what you can then do is take your X-ray information and try to place that on top of a structure that's already been determined, right? And see, because they're homologous, you would expect that there would be some relationship in the overall structure of the protein. So molecular replacement, I don't want to say would be an easier method, but in theory would be a little bit less complicated than multiple isomorphous replacement, which would require you to soak a crystal and then do the X-ray crystallography again. So, just to conclude this little section, nuclear magnetic resonance, X-ray crystallography, they're not really the simplest things to do, but when they work, they can give you amazing information about the structure of the protein. And when you know what the structure of the protein is, you can start to guess and you can start to work towards understanding how that protein functions and what it interacts with. Okay, so let's say you have your protein of interest, you have its structure, right? And now you think that you know of another protein, or let's say you want to find out if there's another protein that it interacts with, right? So one of the things you can do to determine protein-protein interactions in vitro is biolayer interferometry, okay? I think at the beginning of this presentation I said we were talking about surface plasmon resonance. We're talking about biolayer interferometry, so sorry about that. But nonetheless, you can use biolayer interferometry for this. Now, to make things relatively simple, in biolayer interferometry, you have two reflective surfaces. You have one reflective surface that you can call your reference layer, and you have another reflective surface that is your protein-bound layer. And so one's red, one's blue. And you shoot white light at this, and what happens is the white light goes through. The first surface reflects the white light, and also the second surface reflects the white light, right? What you can see is that they form an interference pattern, okay? Now, if your protein of interest is bound to its interacting partner, let's say another protein that you're running on top, right? the interference pattern will change. You can notice that the interference pattern here and here is different. So the way it's reflected from the second surface is slightly different. The phase is different here when compared to here, right? So that gives you an indication that your protein is bound to something. So let's say I have a crude lysate, so I sonicated my cells. I just have all this protein stuff in it, right? Just a mixture of proteins. And I want to see if something's going to bind to my protein of interest. I can run all that through on top of my protein that's been stuck to this reflective surface, the second surface, and then do a couple of rounds of washes. Now, if any of the proteins in the mixture that I added are able to interact with my protein of interest, I will see an interference pattern change, and that will suggest that there's an interaction, right? When we go downstream of that, we can run a SDS page gel, maybe do some mass spectrometry. We're not going to talk about that here. I think we're talking about that tomorrow, actually, and identify what the interacting partner is. Alternatively, of course, if you know, or if you think you know what the interacting partner is, you can actually purify the interacting partner protein and just add that protein alone on top of this, right? So you've got your immobilized protein, that's your protein of interest, and you've got this protein that you think interacts with it. You can add that on top and see whether or not there's interference pattern change. If the interference pattern changes after you wash this, if it remains changed, then that's telling you that there's an interaction. And in fact, if you continue to do multiple washes, what will happen is that the bound protein will slowly come off, and that can give you information about the affinity of the interaction between the two proteins. So it is KD values for those of you who do biochemistry. Another method that you can use to, well, I guess you could use it for protein-protein interactions, but it can also be used for protein-ligand interactions. So if there's something smaller that interacts with the protein, is isothermal titration calorimetry. And many people consider this to be the gold standard of being able to determine affinities, KD values, on-off rates for protein interactions, and protein-ligand interactions, so on and so forth. Now, I guess isothermal titration calorimeter is inside what is considered an adiabatic jacket, which means no heat is lost from the system, okay? And the idea here for you to think about this is you have two cells. You can imagine them as two test tubes, make your life easier. So number one, number two, right? One of them is your control cell. So let's say you're just going to have, let's say, buffer in it all by itself. And the other one will have, let's say, buffer and your protein of interest. In fact, actually, you probably have buffer and your protein of interest here too, just to make the control better. But that's fine. So you've got your control cell here, and then you've got your protein of interest here. Now, let's say you have a ligand that you think is interacting with your protein, okay? If you start titrating in the ligand, so you're adding it in spurts, right? When your protein interacts with a ligand, it will release energy. It's an exothermic reaction because your protein wants to interact with its ligand, right? And that release in energy is sensed by the calorimeter. And what the calorimeter tries to do is make the reference cell increase in temperature so that it is the same temperature as your experimental cell, let's say, okay? So what that means is, as you're adding in ligand, every time ligand is added in, your protein binds to it, it releases some energy in the form of heat, and to maintain the two cells at the same temperature, the heat increases in this cell, right? So the concept here is that every time the heat is increased in this cell, that's recorded, and it's telling you how much energy is being released every time you're adding in your ligand. And eventually what's going to happen is the more ligand that you add in, the more you're going to saturate your protein of interest. And at some point, when you completely saturate your protein of interest, no amount of ligand will increase the temperature anymore. So what you usually end up getting, if you follow the little laser pointer I have, is something that kind of looks like a sigmoid curve, right? So what happens is initially, or maybe not so much a sigmoid curve, but it's kind of like a log. It kind of looks like that, right? And the enzyme kinetics stuff is going to talk about this later on, but you end up getting a curve that looks kind of like this. Okay, and what that means is initially, when you're adding your ligand, protein binds to it very, very quickly. Your protein binds to it very quickly. There's a lot of energy being released, but then eventually kind of plateaus. And it plateaus because every protein now is bound to a ligand, and there's no excess protein, so it's not going to bind to any more ligand. And this information, of course, you get two things. Number one, you get an understanding of whether or not there's an interaction. Number two, you get information about affinity, KD value, and that's really important in terms of enzyme kinetics. Okay, and I know that I haven't talked about enzyme kinetics yet, but that's going to be the next slide. So for the enzyme kinetics sections, I urge you guys to go ahead over to Jove. It's free to make a login, and you can watch this video. It's an excellent video that's going to describe your, I guess, just give you an understanding of enzyme kinetics. One of the things that I was actually just talking about in the previous slide, this is what I meant by you can get a graph like this from an isothermal titration calorimetry. This isn't actually the raw data that you get, but you can get a graph like this from the information you get on isothermal titration calorimetry. And you can see that, the idea here is that as you're adding disregard the X and Y axis here, I'm just trying to give you an idea of what it would look like on ITC, which is what isothermal titration calorimetry looks like. But as you're adding your ligand, you'll see that there's like a spurt of energy release, right? There's a lot of energy release because all these proteins are trying to bind the ligand that you're adding in. But eventually what happens is you're saturating it, and the proteins no longer have any excess ligand to bind to. So you don't end up seeing as much of a temperature change, assuming that this would be temperature, which it's not in this case. So yeah, I think you guys should definitely go over to jove.com, make a quick login, and watch this enzyme kinetics in vitro video. It is an excellent way for you guys to understand kind of what the old school method of understanding enzyme kinetics is, understanding the on-off rates of your protein interacting with your ligand, affinities, and so on and so forth. And with that, I think that kind of concludes the presentation for today. It's very long. I kind of expect most of you to just kind of go to different sections of the video and focus on the things that you're really, really interested in to try and understand exactly what I was saying. Obviously today we've talked about amino acids, peptides, protein structures. We delved in a little bit more detail into CRISPR-Cas function and genome editing, and then I gave you a relatively surface overview of protein purification analysis. I did not go into too much detail. I did try to simplify things a little bit, so hopefully I provided sufficient information for a lay audience to understand what it is that we were talking about, but at the same time maybe provided some details that somebody who's a little bit more versed with this field still gets something out of it. Thank you very much.